Okay, so in this video, we will discuss the next topic in Cycas, that is the internal morphology. So we had already discussed about the external morphology of Cycas. Now we will take a look at the internal morphology of the root, the stem, and the leaf. Okay, so we will start with the root. So now here in the root, the internal morphology of the root, the young root of Cycas, is somewhat similar to that of a typical dicotyledonous angiosperm. And as you can see here in this picture, this is the diagram showing the transverse section of young root, and it shows a nearly circular outline. Right, it has a nearly circular outline in both these pictures, as you can see. And the regions which we can see here, the first region, right, you can see here, this is the epidermis. All right, so the first region here in the root, when you take a look at the TS section, you have the epidermis, and the outermost single layered epidermis, as you can see here, it is referred to as the epiblemma. And this epiblemma, it consists of thin-walled parenchymatous cells. Okay, that is the soft tissues, and that is the cellular tissue. They are parenchymatous, meaning they are made up of soft tissues, and a few cells of this layer, here it is not showing, but few cells of this layer, sometimes they elongate to give rise to root hairs. Alright, so even root hairs are present in the root of the cycas. So coming to the next layer, here you can see this layer. Alright, it is the cortex. Now the epidermis is followed by cortex, consisting of thin-walled parenchymatous cells, and these cells of the cortex, they are actually filled with the starch. Okay, they are filled with starch grains, and the starch grains, they are scattered everywhere in the cortex. So scattered in the cortex are also few dark brown tannin cells. So remember, in the cortex there are starch grains as well as tannin cells, and also in the cortex there is presence of the large number of mucilage canals, and each mucilage canal, it is bounded by an epithelial layer, or what we call secretory cells. And the secretory cells which are present here in the cortex, they are filled with the mucilage. So next to the cortex comes the endodermis. You can see here this layer. Alright, the first layer here is the endodermis. Now the endodermis, as you can see in this diagram, in this picture here, the endodermis is actually single layer, and it is also distinct. The cell possesses characteristic Casparian thickenings. Okay, so here in the endodermis, in this diagram you cannot see, but then just remember that in the endodermis we can find the Casparian thickenings. Okay, that is those thickenings, band-like thickenings, which are usually present in the center of the root endodermis. So these types of thickenings are found in the endodermis. And next to, just below the endodermis, alright, this layer here, just below the endodermis here, alright, this is the pericycle. Now the pericycle is actually multi-layered, consisting of thin-walled parenchymatous cells, and just like in the cortex, even the pericycle here, it is filled with the starch grains. So just below the pericycle, we can see the vascular tissue. And here the xylem and the phloem, you can see these are the phloem, alright. The xylem, you can see this is the secondary xylem, this is the phloem, and this is the metaxylem. Alright, this one dark here in the middle, it is the metaxylem, whereas these are the primary xylem and or the protoxylem, and this is the phloem. So the xylem and phloem strands, they alternate with each other and show a radial arrangement. The roots here in case of Cycas, they are usually diarch to tetrarch. That, as you can see here, diarch. Why? Because two xylems can be found. So in this case, we can see two xylems. Clearly in this picture here, in this first diagram, you can see, okay, these are the primary xylem. Sorry. This is the secondary xylem, and these are the phloem. Okay, so you can see here they are diarch, that is having two xylem groups, but some Cycas species they even have four centripetally developed xylem plates. So if they have four xylem plates, they are referred to as tetrarch. And some species of Cycas, they are polyarch, meaning they have many xylem bundles. And you can see here that the xylem, in this case, it is also known as exarch. So what do we mean by exarch? You can see in this first figure here, figure A, you can see, or you can see even in the figure B, as you can see that the metaxylem, alright, the metaxylem, it grows towards the center. So you can see that the metaxylem here, it is towards the center, whereas the protoxylem is growing towards the periphery. Okay, so you can see the metaxylem in towards the center and the protoxylem towards the periphery. That is why these type of xylem are known as exarch. And the tracheids, they have the tracheids here, that is the water conducting cells of the xylem. They usually have the they have spiral and scalariform thickenings. Okay, that is the xylem vessels, they are arranged, they have thick bands which are arranged in the form of scalariform cells. We can see that they are arranged just like the rungs of the ladder, or they also have these reticulate thickenings. Reticulate thickenings when the xylem vessels, they are arranged in the form of a network. Okay, in the form of a network of the lignin deposition. And here in the whereas those, just remember that the tracheids, that is the water conducting cells of the xylem, they have spiral and scalariform thickenings in the protoxylem. Okay, in the protoxylem, whereas in the metaxylem they show reticulate thickenings. So just like in the form of a network they are arranged, and whereas the vessels are absent. And the center of the stele, that is the stele, the center core of the stem, of the root here, the center core, that is known as the stele, it is occupied by the metaxylem. Alright, as you can see clearly in this figure B, that they are occupied by the metaxylem. This core here, this middle portion, is known as the stele. It is occupied by metaxylem, or sometimes metaxylem mixed with parenchyma. And the phloem, it consists of sieve tubes, that is the living cells of the phloem, which helps in conduction of food transport. Okay, for food transport, especially the sugars, and also consists of parenchyma. So the phloem, it consists of these sieve tubes and parenchyma, and the older parts of the roots, they show the presence of bast fibers in phloem. So in phloem, okay, this is actually the picture showing the TS of a young root. So you cannot see here the bast fibers, but they are usually present in the older parts of the roots. And these bast fibers, they are plant fibers which are present in the phloem. They actually support the conductive cells of the phloem, and they also provide strength to the root. And the pith here in this case is reduced or it is absent. So this is the TS section, or the internal morphology of a young root. So in the next video we will see the secondary growth. Okay, the secondary growth which takes place in the root of cycas. In here we are talking about the normal root. Remember that we have two types of roots, the normal root and the colloid root. So the colloid root, I will talk about this later. We will discuss now this secondary growth. This growth here with which I am showing the internal morphology is of the normal root.